I think first is a bit of a disclaimer, and also for me to a starting point. I'm not a virologist. I think for me, the important part is understanding process in the cell upon any kind of different stresses. And I think for me, one of the main focuses has been stress in different cellular compartments, also upon protein misfolding, but also extracellular stresses, such as a pathogen infection, for example. And I think what has really been important is trying to kind of integrate these stresses and to somewhat get towards more of what I would call a molecular systems biology or a systems medicine view of what happens in the cell upon these stresses. So integrating different processes such as the signaling events that are occurring, and transcription and translation changes, and of course also the outcomes that go along with that, with effects on cell cycle and proliferation or metabolism. And for me in particular, one of the things we've been focusing most on in the last years has been there on translation. And I think I'll give you one kind of problem, a technical difficulty where we were stuck, and I will explain this technical parts to you because that's the method that's later used to study SARS-CoV-2 infection in cells. So I in particular was stuck at this point where there's a lot of stress responses, and for example, mTOR inhibition upon stress in the cells, where we know that there's really severe attenuation on translation in the cell. In this case, about 60 to 70%. Just want to mention this is a Nature paper from 2012 from the Sabatini lab. And if you look at that by polysome profiling, you also see the same effect. Translation is largely gone. However, if you then use the standard method that I think is most commonly used also in my lab to look at translation and ribosome profiling, you see that there is a normal distribution around 0. So what that essentially means that from here, you cannot tell that there's overall an effect on translation. So it does not really affect this here. This is not a mistake. So this is really, I think, one of the technical limitations in these methods where there's a normalization bias going on. And we really wanted to understand what happens upon stress, what happens in infection to translation. And we needed to get a picture where we can get that resolved on a per gene, per protein level, but really where we also see this decrease in overall translation levels. So my lab heavily focuses on using proteomics approaches, and that's what we turn to. And of course, for many years, there have been these approaches using pulse silex. So the idea is there you have kind of a cell growing in a normal culture. It's all light labeled, and you can then change the medium to heavy lysines, heavy arginines, and you can monitor over time how these heavy amino acids are getting incorporated into the proteins. So you can follow this profile for individual proteins over time and determine the translation rates for these individual ones. The real problem is if we want to look at acute effects, so within minutes or hours after an infection, after treatment, after stress, we're kind of here in this window. And this has been technically really difficult because essentially we cannot measure most of those signals. So we're stuck in a situation like here with dynamic silex, where in most instances the light peaks are so tall and the heavy peaks so small that we cannot select those for detection, so we do not get any quantitative information. So essentially, it doesn't work in these instances. And where they have been very useful is more long-term labeling, let's say for a day or even longer. And what we came up with is kind of, I think, is a good solution for that. It's a method we call MEPROT, or multiplex enhanced protein dynamics, where, and I will stay away a bit from the technical details, so we use a tool, it's called tandem mass tags, that allow us to multiplex samples in proteomics, similar to what we do in next generation sequencing. And what that allows us to do is to include one channel that is what we call a boost channel that is fully heavy labeled and pushes the signal for the other 9 samples in the peak up here. So we have a big enough peak now that becomes detectable. We can analyze them in the machine. We can then tell how much of the intensity was there for the 9 individual samples in one of those 11 plexes. We can do that the quantification relatively to the boost channel, and can even determine noise channel with this baseline approach. And this has worked really well. I want to mention here that this was worked by a brilliant PhD student in my lab, Kevin Klum. And it also works in cell. Here's an example from HeLa cells where we're labeled from between 15, 30, 60, and 120 minutes. And as you may appreciate, we do get very tight clustering of the replicates and the linear correlation. So across the whole range from short to longer labeling, we are able now to measure these newly synthesized proteins. And mind you, here we're going down to less than 1% any given protein to be labeled. So it's really low, but with this method now, with this boost, we can get there. And we apply this, for example, in this example that I gave before, you looking at mTOR, but also, for example, at EF to alpha phosphorylation and integrated stress response. And now I think we get much closer to what we would expect. We do see this global reduction in translation, about 60%. And now we can determine for every single protein here in this experiment goes over 5,000 proteins quantified. For every single one now we can determine how does translation change. Is it going down? Is it going up? And so on. And because it's essentially largely kind of a typical mass spec approach, it's much easier in the throughput and in the sample amounts required than, for example, ribosome profiling. So we can do really big experiments. Here's 27 different samples over 5,000 proteins quantified to look at specific questions. And the one we answered in this paper looked at is really whether there is an overlap between mTOR signaling, so here on the left, and the integrated stress response. And indeed, it was. So let's summarize the technical part. And there we have this new approach now where you can have up to 9 samples in a multiplex. It's actually extending at the moment. We need very few cells, much less than what we need for ribosome profiling, about 100,000 cells per sample, and it costs us about €400, $450 or so. So it's much cheaper than ribosome profiling, and our current record is about 8,000 protein quantified. So with that, we're back at that stage, and I told you that we're interested in stress responses, and we applied these methods to viral infection, bacterial infection, because also I realized that there's a real niche, and our understanding of the translation control after infection is still poor. And then this happens on the beginning of February. I mentioned before, I'm located in Frankfurt in Germany, so we have one of the biggest airports in Europe, and typically if anything, a pathogen arrives in Germany, or often in Europe, it arrives via Frankfurt, and that happened here too. We had an evacuee flight from Wuhan in China on February 1st that came to Frankfurt, and then amongst the people who were evacuated, 2 were actually infected with SARS coronavirus 2. And the Virology Institute here in Frankfurt managed to extract viral isolates from those 2 people. And then we really very quickly got to collaboration, and that was again with Kevin Klein in my lab, and especially Denisa Boykova and Benjamin Koch under the guidance from Professor Linders Sinabel at the Virology Institute in Frankfurt. And what they managed to do is really within a couple of days to set up a cellular infection model for SARS-CoV-2. So what they used is CACO2 cells with colon endothelial cancer cell line that had worked really well in the past to study SARS infection and to have a cellular system for that. And you can see here how that works. So if you have those cells growing with a mock control, they look intact. However, after 24 hours of infection with SARS-CoV-2, you see a clear cytopathic effect. The cells were infected, and they're starting to break open and release virus. And that we also validated here on the right using qPCR assays to look at the release of the viral RNA into the supernatant. And you can see over time that there is this release of virus. And then we use this method that I mentioned before, this MEPROT, to the time resolved manner over time look at changes in translation and also in the total proteome upon infection with SARS-CoV-2. So here's we had these CACO2 cells and infected them with this isolate. And then after 2, 6, 10, and 24 hours, we always pulse labeled for the same amount of time, and then harvested the proteins to look at total proteomes in translatome. And just a short disclaimer, and I think this is really something that's become really important in the times of Corona. All the data that I'm showing to you now, it's a published paper, but I also try to make sure that people have very easy access to the data. So I put up this special section on my website where you can find the raw files, the labeling schemes, and so on. In the paper, that we also put supplementary tables with all the proteins, but I also set up a kind of simple interactive online tool where you can just type down the gene name, protein name of interest, and you can see what were the changes that we measured. So if we now look the actual data, that's where we were. This is a PCA plot to just get an overall idea of what was happening after infection. And the first good thing to see is that replicates clustered together fairly closely. So that's good from a reproducibility point of view. But we also saw that there were really changes starting at mildly at 6 hours after infection, but especially after 10 hours and 24 hours. We really had huge rearrangements in the cell after infection. And here's a view of what we saw for 5 different viral proteins that we detected. So this is changes in translation. And as it should be after an infection, you can see that all those 5 proteins become translated after the infection. As I mentioned before, the labeling times at all these time points are always the same. So this continuous increase that we see for these proteins is not just an accumulation. It actually means that it's later and later time points, there's actually more and more translation happening of viral proteins. And there it was, for us, really one of the big surprises when we then looked at the host cell proteome and what happened globally at translation. And I think you may appreciate from here, there's overall not much happening at all. So we do see a reduction of a maximum 23% after 10 hours of infection. But this was really not as severe as people had suggested for other viruses. That was a bit surprising at that point, and I think the stage we are at the moment, one of the limitations is that I think we need to get a better understanding of infections with other viruses, such as SARS and MERS, but also unrelated viruses, to get a better idea how severe the reduction in translation actually is upon infection and how that compares to SARS-CoV-2. Because a lot of the studies that have been done were really upon overexpression of single effector proteins from a virus to see what that does on translation. And we then looked in a bit more detail what changes in the cells, in the host cell, what is increased. And I think there we probably saw a bit of an explanation why the translation rates keep rising for the viral proteins, even though there's no shutdown of the host cell proteome translation. And is that we saw really a lot of terms around ribosome, translation, elongation, and so on that were increased. So that means that the translation machinery is increased upon infection. And then we were just wondering if we perturb translation in the cells upon infection, whether that could affect viral replication. And indeed it did, using drugs like cyclohexamide and emmetine, both blocking cytosolic translation. You see here an inhibition of viral replication at below micromolar IC50s and at concentrations that were non-toxic to the cell. We then also looked at the whole cell proteome to see what happened there in the host. There were really 2 main clusters, as you would typically expect, one that's increased, one that's decreased. For us, the interesting part was really the increased cluster. And as you may see from here, this is the site where we looked at increase in proteome upon infection. This is in triplicates, so it's really mainly this 24-hour time point where we saw really severe increases in host cell proteome proteins. And there's 2 really main pathways that really struck us. So we're not so much interested in finding a single protein that goes up or that goes down, because there can always be some variation in the system. And also, if you look at these really dynamic systems, it's sometimes hard to get very kind of strong statistics also behind it. So what I think is much more interesting, and that goes back to this systems biology approach, is to look at all pathways that are changing. You're not finding one protein that changes, but 5 that are connected and have a similar function in the same network. And there what was really sticking out is changes in the spliceosome, splicing overall, but also in carbon metabolism from TCA cycle, metabolism, carbon metabolism, and so on. So we wonder whether there would also be kind of a therapeutic window in those cells to see whether it can affect viral replication. For that, we tried first a spliceosome inhibitor as a that targets SF3B1. It's called Lydian Light B. And part of the reason for picking that was also there's not that much known on what happens to the host cell's lysosome upon infection, whether it's required or not. However, there had been a lot of papers describing that overexpressed viral proteins do interact with the spliceosome. And it wasn't so clear whether that has a biological meaning in the end for the replication. What we saw with the Lydian Light B is here, it's a really very low nanomolar concentrations. We see an inhibition of viral replication. Again, these were conditions that were non-toxic to the cell. And then we also addressed the carbon metabolism that I mentioned. We're using 2-deoxyglucose, which would inhibit hexokinase, or the first steps, using glucose in the cell. And we saw, again, inhibition of viral replication in the cells. And that was kind of nice because there had been studies already out there, particularly on rhinovirus, where also in animals, groups had shown that there's a therapeutic potential using these compounds. And there's actually also companies that have been trying to develop prodrugs of 2-deoxyglucose in cancer therapy, and they're really heavily invested now in trying to bring that to the clinics and to generally assess the efficacy of those drugs and how what concentrations or amounts you can give it to patients in theory and so on. And then we went to the next step to really take the information that we got from the overall abundance changes in the host cell proteome and to look at profiles that kind of mimic what the viral proteins do. So here on the absolute protein amounts or abundance levels, we could detect those for 9 viral proteins, and then we followed which host cell proteins follow a similar profile. And this is really the network of what was sticking out, and I think not necessarily extremely surprising, we found that there's a huge variation in nucleic acid metabolism. Of course, RNA virus, it needs bases just as any other virus to reproduce its genome, and these need to be replenished. And there we tested now one compound, ribavirin, which is an IMP dehydrogenase inhibitor, and we found, again, at micromolar concentrations in IC50, inhibition of viral replication. I think it's not a general feature that if you interfere with that system, you automatically get that effect. We also tried a lot of pyrimidine inhibitors, for example, or inhibitors of pyrimidine biosynthesis. And we never got this clear inhibition of viral replication. But this particular ribavirin and how it can be used for us has been kind of a very nice story. As I mentioned, biologists or basic researchers usually don't necessarily produce results that can be directly translated to the clinics. But I think here is one of those examples where this data has helped, and there's a clinical study now ongoing in Canada where Virusol, which is an aerosolized form of ribavirin, is now being tested in COVID-19 patients. So the idea is there you could spray it directly into your lungs with the hope of suppressing the viral replication in your body. So the point at which we were there is that we had this situation where we had established this cell culture model for SARS-CoV-2 infections, and using whole cell proteomics and translation proteomics, we made different observations. One is that the global host cell translation largely remains intact, which I think has a lot of implications. Also that the viral proteins have to compete with the host cell proteins for translation. So there need to be alternative ways to make it possible to produce these viral proteins at these higher rates that are required to produce virus. And part of that might be driven by upregulation of the translation machinery. But we also saw a couple of pathways in the cells that were upregulated. I mentioned some of them. There was translation, nucleic acid biosynthesis, splicing, proteostasis, and glycolysis that were all increased upon infection. We assumed it may have an effect on SARS-CoV-2 replication in the cells, and indeed, inhibitors for these different pathways individually all led to inhibition of viral replication. But I think the point we're at then is that we now identified these pathways and that there is this connection between these pathways and replication, but we kind of had no understanding on how we get there. So essentially, these gray arrows that underlie the signaling that is required towards replication, we could not understand. This is something, a piece of information that we cannot get from the data that we had at this point. So we went further, and again, employing
Christian, your 2-minute mark.
Okay, thank you. So again, employing this cell culture infection model, we now turned to phosphoproteomics to gain an understanding of signaling changes. So there we infected cells with mock in 5 replicates, or with SARS-CoV-2 also 5 replicates, pooled them together. We're still a bit limited by how much material we can get, so we really had to go this way, multiplex them and shoot the total proteome and look at the phosphoproteome. It's very little we get out of those 10 samples, so we couldn't do much fractionation, but on the positive side, even from this little amount of material, we managed to quantify over 15,000 phosphosites. And here's shown the changes in the whole cell proteome. We discussed it already, so leave it out. But there were really severe changes in phosphorylation in the cells. And that was mainly on serine, partially driven by the IMEC method we used to enrich those phosphopeptides. And just to mention this point, so this data is now on bioarchive. So again, we made Excel sheets available for this data. and also the raw files. And then we first looked at phosphorylation of viral proteins, and a bunch of them that we could detect. I think here, one of the difficulties is that we still, a lot of them don't have a full understanding how they work, what they do in the cells. So we also cannot tell all of them what this phosphorylation means, but we identified them. What is striking is that for this nuclear protein N, for which for part of this RNA binding domain here, there's a structure available, and the 3 phosphosites we detected in there are all on the same surface. I think strongly suggesting that there might be a motif around there or some interaction surface that could be affected by these modifications. And then we tried to gain an understanding what are really the key pathways in the cells that are regulated. And at the same time, we also wanted to see and identify pathways where we would have a good chance to affect them with pharmacological inhibitors. So we're looking at proteins for which a phosphorylation increase was there that was not mimicked by a protein increase, but by real increase in the signaling going on. And that's network over here overlaid with a network of drugs with an FDA, with a network of drugs that have been FDA approved. And I think quite striking, very clear in the middle, it all resolved about EGF receptor, and so growth factor receptor signaling. And this is what we saw actually for different growth factor receptors. We saw a severe increase in their phosphorylation. So this is this red colors. Every part of the circle is a phosphorylation site, but also of the downstream signaling cascades, RAS-RAF signaling, AKT, mTOR, and so on. So really wondering whether this is something that's important for the virus, whether this is a key biology that could be targeted. And so we then mentioned there are different growth factor receptors, and we thought this could be different for different receptors, might not be good to target them, but the downstream signaling is very similar. So we picked out these 5 different compounds that inhibit downstream signaling of growth factor receptors at different points. And all 5 of them prevented viral replication in the cell culture model at low IC50s that were all within the range of what can be achieved in people. They also prevented release of viral RNA copies into the supernatant, and they also prevented, which I'm not showing now, expression of the viral proteins that we looked at by staining for viral proteins in the cells. So as a summary, I mentioned to you that I developed these new MEPROT methods to look at acute translation change in the cells. We set up cellular infection systems for SARS-CoV-2 infection, and we identified a range of pathways that are upregulated upon infection and inhibitors for which prevent viral replication. And we then turned towards the signaling part of it, looking at phosphoproteomics and identify the growth factor receptor signaling inhibition and prevent SARS-CoV-2 replication in cells. So with that, we'd really like to thank my group. Fortunately, we cannot take updated pictures anymore because we're not allowed to meet in the current crisis. There's really a lot of people strongly involved, especially Kevin Klang and Georg Tascher, also others here involved that are supporting this. The collaborators I mentioned also with pictures, including Sandra Zizek and my mentor in the department, Ivan. And thanks to the different funding sources and for you for your attention. And really looking forward to the questions.
Thank you, Christian, for a very exciting talk. While we're waiting for people to raise their hands, let me ask you a question. So we've been doing similar studies, again, using sort of systems approaches to study how the virus induces changes in the host cell. And what we've seen when we applied these methodology to different cells that were infected with the virus, including bronchial epithelial cells and several cell lines from cancer, is that the results were highly dependent on the context. So you're using CACO2 cells, and those are colon adenocarcinoma cells that have essentially expressed the ACE2 receptor. So is it possible that, for instance, the involvement of EGFR pathway and cascades downstream of those is maybe a byproduct of the fact that you're using a cancer-related cell line where we know that EGFR signaling is a particularly relevant mechanism of activity?
Yes. I mean, as a general answer, yes, it is possible. And I think it's actually probably part of the pathology because now we know that there's so many different organs that can be infected. And I think the effects that the virus will have on these different organs will actually be different. Right? I think for us, the limitation was, and again, as a non-virologist, something to learn that it's actually difficult to find permissive cells. And it took quite a while to set those up. I mean, especially the first study, we sent it out for review in the end of February. So at that time, there was just simply nothing available.
Yeah.
I think now we're getting much more to the point where we have primary cells. We tested it. It's kind of interesting and also boring because we tried out primary lung cells from people also in differentiated form, and we see a very, very similar response. And so it's kind of encouraging because it's kind of recapitulates what we saw and then confirms it. But
Yeah,
there's
but
not.
so it would be interesting for you to compare your methodology versus on CACO2 versus CALU2, because those are the sort of the lung adenocarcinoma versus the colon adenocarcinoma. Both of them, those models are available from the literature, and we see very different activation. So it's actually reflecting how the infection may really do different things in different parts of the body, but also a little bit worrisome because we don't know whether the results that you get from one context are going to effectively prevent infection and replication in a different context.
Absolutely. And I think for us, I mean, we're trying to move completely away from cancer cell lines, also CALA2, really trying to get to primary cells. The problem is for any primary cell you try to set up, you have to reestablish the infection kinetics because it's just different for every single one.
Yeah, no, it's a lot of work.
Make comparisons very different because you don't know whether the difference is just because you hit the cells at a different time point of infection because it just takes longer in that cell line.